All right, welcome. So, think about this for a second. Over here, you've got the world of biology, cells, DNA, all that complex stuff. And way over on the other side, you have the world of artificial intelligence. Now, on the surface, they seem like they're from completely different planets, right? Well, today, we're going to dive into a really powerful mathematical tool that can find these incredible hidden connections between worlds just like these. A tool that aligns data even when they speak completely different languages.
So that's the big question, isn't it? How do you actually align data from different worlds? I mean, how do you find meaningful connections when you can't just slap one data set on top of the other, when there's no shared map, no common grid to guide you? This is the core problem we're going to unpack today.
Okay, let's make this real. In the world of single-cell genomics, there's a tricky problem. A lot of the time, the act of measuring something actually destroys the cell. So what does that mean? It means we can take a group of cells, measure gene expression in one half, and then in the other half, measure how the DNA is packaged. That's called its chromatin accessibility. But here's the kicker. You can't get both of those measurements from the same single cell. You end up with two completely separate data sets that are describing the same biological reality, but there's no direct one-to-one link between them.
And that, right there, is what we call the unaligned data problem. It's this situation where all our standard tools for comparison, the ones that need a shared frame of reference, they just don't work. They completely break down. We need a totally new way of thinking about this.
The key term you're going to hear is heterogeneous data. Just imagine one of our data sets lives in a gene world, where everything is defined by gene activity. The other one lives in a DNA structure world. You simply can't measure the distance between a point in the gene world and a point in the DNA world. They're just incompatible. In math speak, they exist in incompatible metric spaces.
So, how on earth do you build a bridge between these different worlds? Well, this is where a truly brilliant mathematical idea called Gromov-Wasserstein, or GGW, steps onto the scene. And it offers a completely new, kind of mind-bending geometry for comparison.
And this slide really shows the genius of it. See, on the left, you've got your traditional methods. They need that shared grid, right? A common coordinate system to match points directly. But what if you don't have that? That's where Gromov-Wasserstein's on the right is so clever. It just completely ignores the absolute positions. Instead, it only looks at the shape of the relationships inside each world. It asks a different question. Is the pattern of distances between these points here in world A similar to the pattern of distances over in world B?
So this is the crucial point. This is the revolutionary shift. We stop trying to match individual data points based on what they are, and we start matching them based on how they relate to all their neighbors within their own world. It's amazing. We're not aligning points anymore. We're aligning the shapes, the internal geometries of the data. So this is this powerful, elegant, beautiful idea. And it sounds like we've solved it, right? But as so often happens in science, this brilliant concept ran headfirst into a massive, and I mean massive, wall. The promise here was huge. We're talking about a potential universal translator for data, a way to connect any two data sets, no matter how different they were. But there was a catch, a really, really big one.
The problem is NP-complete. Now, if you're in computer science, those might just be the two scariest words you can hear. It basically means the problem is monstrously, ridiculously difficult to solve exactly.
And this quote really gets to the heart of it. See, finding that perfect optimal alignment is what's known as a quadratic assignment problem. And what that means is, for every potential match you try to check, the complexity of the problem just explodes. It's a total computational nightmare.
So what does NP-complete mean in the real world for you and me? It means that the exact solvers could only handle teeny tiny data sets. We're talking just a few hundred data points. For modern genomics or AI, where data sets have millions or even billions of points, that's completely useless. The brilliant idea was, for all practical purposes, dead in the water.
But of course, the story doesn't end there. An elegant mathematical fix came along that completely changed the game, and it turned this impossible problem into an incredibly practical tool. This was the entropic breakthrough.
The solution has a cool name: entropic regularization. The best way to think about it is like adding a tiny bit of blur or fuzziness to the alignment. So instead of demanding a single perfect one-to-one match for every single point, it allows for a softer, more probabilistic matching. And this one little change has a massive effect on the underlying math. So let's just walk through how this magic trick works. You start with that tangled, impossible NP-complete GW problem. Then you add this clever mathematical smoothing term. And all of a sudden, the problem's landscape just smooths right out. making it actually possible to find a solution. This opens the door for these incredibly fast algorithms to find a really, really good approximate answer.
And what you get at the end is Entropic Gromov-Wasserstein, or EGW for short. It's a method that is not only fast enough to run on these massive modern datasets, but it's also statistically reliable. It's this fantastic proxy that perfectly captures the spirit of the original GW idea while actually being achievable in the real world.
So, with that computational wall finally torn down, EGW has just unlocked this huge wave of innovation across a whole range of fields. I mean, this isn't just theory anymore. This stuff is having a massive real-world impact. And the applications are just incredibly diverse. I mean, of course, it's solving our original biology problem in single-cell science, but it's also being used to align different AI language models to see how their internal structures compare. It helps computers with 3D shape matching. It figures out how complex networks or graphs correspond to each other, and it even helps generative models create brand new, coherent data. EGW is really becoming a foundational tool for data integration.
And let's bring it all the way back to where we started, back to ourselves. For our single-cell data, EGW gives us this, a probabilistic coupling matrix. It's basically a heat map. What it tells us is that, hey, cell number 1 from our gene expression world is, let's say, 70% likely to be the same as cell number 3 in the chromatin world, and maybe 20% likely to be cell number 4, and so on. It's that soft, fuzzy, beautiful alignment map that we needed all along. So EGW has been a total game changer, no doubt about it. But the work is, well, it's far from over. The frontiers of data are always expanding, and researchers are pushing these alignment tools to their absolute limits to keep up.
You know, the challenges just keep getting bigger. Datasets are growing in size and complexity at an exponential rate, and the demand for even faster, even more robust alignment methods is constant. This is a very, very active area of research right now.
So what's coming next? What's on the horizon? Well, people are developing neural solvers that actually use deep learning to scale EGW to absolutely massive datasets. They're creating unbalanced versions for situations where the datasets don't even have the same amount of stuff in them. And really critically, they're working on formal guarantees to make sure these algorithms aren't just fast, but are provably reliable.
So, we've seen how this brilliant, powerful idea for comparing different worlds was trapped by computational limits, only to be set free by an elegant mathematical trick. And that journey, from an impossible problem to a practical tool that changes everything, that's what drives science forward. And it leaves us with one final thought. From biology to language to art and beyond, our data worlds are constantly multiplying. So the question is, with tools like EGW, what will we be able to align and to understand next?